that. And then just for anybody that not familiar with who Carson is, he hails from Oregon, as he alluded to earlier. He's been into carnivorous plants since he was about 6. And due to his geography, he's up there with Sarracenia Northwest. He was introduced to the wonderful world of carnivorous plants from Jeff. And as an adult, he went to go work for them for about 3 years. And currently now he is pursuing his horticultural degree at Oregon State University. And I actually met with Carson. I say that loosely. We met online because he was kind of playing around with an idea of getting all the various carnivorous communities throughout the country together and do something on a national scale. And so that's how I got to know him. He's got big ambitions and dreams for the carnivorous community. So pretty cool stuff. I am going to start your PowerPoint. You know what, let me do this. Let me share my screen first. If I can find my, hold on a second. I've got way too many windows open.
I'm enjoying what I'm seeing in the chat box.
Yeah, go through that. Let me, why
Oh yeah, Vancouver water is actually even worse than Turtle Pond water to grow your plants in, Patrick, as we all know. Yeah, we tried doing that, but
all right. So
I know, Patrick. I'm not just idly poking fun at him.
There we go. So I've got the screen up. Carson, whenever you're ready, take it away.
All right. So, and we get to talk about, or I get to talk about, or talk at you, my favorite thing today, which is, you know, when John first contacted me for this, he said, I'd love to have you talk about some sort of Sarracenia identification method, or because, you know, I guess it's pretty easy to conflate a lot of the pitcher plants out there, a lot of the Sarracenia. When you don't know what you're looking at, everything can be a real blur. And I remember when I first started working at Sarracenia Northwest, I didn't like Sarrs that much. And the only thing that got me into it actually was a really, you know, forced but intimate discovery of what those traits actually mean and what they are. And this is not, it was done in such an amateur and business oriented way, but in that business, I had to get it right, or else we'd pay the price. And so I came up with, I actually never came up with a scheme until recently, but over my 3 years there, I became familiar with the traits that would differentiate not just look-alike plants, but which would tell me what a plant could possibly be if it were a hybrid. And oftentimes when you look at a hybrid, you can kind of identify, okay, that might, you can identify by the shape perhaps, or just by looking at it, you know, superficially. Not only does it not look like a species, but it's got something to it, and that something is the thing that I needed to articulate when going from plant to plant in a nursery really rapidly, and amongst a lot of plants that look very similar. So this was my system, and writing this out in a program, and I could have written a book. I had about 4,000 or 5,000 words written down. And I said, this has got to stop. So forgive me if this isn't exactly consolidated, but do know that there's quite a bit to say, at least on my part, about what about my experiences with this, and we'll get to that. So next slide, please. Yeah, there we go. Okay, so there's me kissing the plant. That's how much I love carnivorous plants, man. They are my absolute life. Most of this has been done by, I've been introduced by John. One other thing I think I might want to add is I do grow Sarracenia from seed indoors under lights, as well as outside, but mostly inside. And in doing that, most of my plants are, in fact, all of my plants are genetically different. So what I'm looking at in every single one of my seedlings is a goal, and that goal is traits. That goal is a set of traits that I'm attempting at in crossing two different plants. Next slide, please. I actually don't have control over this slideshow here. It's all on John's end. So I don't know if y'all can see it because my video box here thing is has been, there we go. I'm going to move that to the bottom bar. All right, so Sarracenia have something to discern between our traits, but also anatomical parts. Everybody's got a nose or ears. Very few people are born without one. Some people are, but our genome usually codes for a nose. That's not a trait. The trait is whether or not, the trait that I'm discussing is whether or not that you got a big old nose or a little nose, or big ears, or what color hair you have, or even what color skin. Now in humans, we better not, as the times have been very focused on recently. We should never apply market value to that. But to Sarracenias, in horticulture, that's all we do. It's artificial selection of genetics, and the genetics specifically referring to discrete traits for each anatomical part of a pitcher plant. So that's the hybridization of it. And use in everyday use, what you can do is you can use these traits to discern one plant from another if they look very, very similar, or to discern one plant from another in phylogeny or taxonomy when identifying species. So a good part of Sarracenia botanization, or Sarraceniology, I guess you could say, is the comparison of various traits. For example, if you read, and this book is alluded to in the program, if you read Schnell and McPherson's Sarraceniaceae of North America, pretty much everything they're talking about are the different traits amongst the anatomical parts of plants. That's how significant they are in botany, and it's no different in horticulture. But in horticulture, the only real difference in horticulture is we make money off of it, and we don't care really what the plant's called, just that it looks cool. Another type of trait, besides the morphological, physiological, are phenological traits. And the phenology of Sarracenia are diverse, but almost all the plants go dormant in winter. But when they come up from dormancy, they flower at different times. They produce different sets of pitchers. Some plants produce only one set of pitchers. Some produce all the way up to 3. I've heard rumors of them producing up to 4, but I really don't know that. Some plants keep their leaves for a long time. Some let them die off. Some have Phyllodia, some don't. If you're familiar enough with a clone, you'll be able to pin these phenological traits to that clone and you'll be able to understand how it travels down in progeny. Have I ever done that? No, I've not, and I'm woefully unqualified to talk about this because I've only had 3 or 4 years experience. But in that time, I have learned so much. I've also learned that, there's only there is so much more for me to learn. But I encourage you to approach this as a explorationally instead of dogmatically, because really that's what we're doing, is exploring these plants as hobbyists. Next slide, please. Sorry, I'm not even paying attention to what's on the screen. So I summed this up. Traits will help you articulate the differences in your plants. are amongst a group of plants. Instead of them being a blur, you'll be able to fluently tell what the differences are between not just seedlings, but say you're at the store and you see a ton of different Sarras up for sale. Can you choose the best one? Can you choose the one that you're going to like the most? Can you choose the most valuable one? Can you trust yourself to do that? I don't think I could do that until about 2 years into staring at pitcher plants every day and all day. And now I think I'm at a point where I can, but it takes a while. There is so much to look at per plant, and really, in the end, it's so subjective, because there really is no value on life. The most rewarding thing about a Sarracenia is how it grows, but any sommelier will tell you can drink any wine, but but they like to profess about it. And as a Sarracenia nerd, that's kind of my job. So I have boiled it down to differentiation, judgment, and assessment of status amongst the physiological traits apparent to me, including the phenological. You can tell where a plant's been, how it's been grown, what conditions it's been grown under, right? How healthy it is by looking at the traits from a healthy plant to an unhealthy plant. If you know your clone well enough, or if you know the genetics of that plant well enough, you'll be able to tell by looking at the traits, not just looking at the plant and eyeballing it. There's articulation of that, and regards to genetics and hybridization. In gene play, knowledge of traits gives you insight, foresight, and retrospection. It gives you a window, perhaps, from which to imagine, because all of this is imagination, as is any science. Science, not solving problems requires imagination. It allows you to solve questions to a rough degree, not completely, but allows you to infer the consequences of a cross. It allows you to imagine what could be inside of a cross if you know the relationships between, if you know your traits well enough in the parentage. It allows you to plan crosses, and in planning crosses is probably the most intensive use of trait knowledge. Phil Felici plans his crosses 10 years in advance, and it pays off, and he knows his traits. Next slide, please. And there's trait unreliability, right? You're not going to be able to see every trait or the distinguishing traits in your plants every year. Traits will yet exist, but maybe not the ones that you expected. So you can get a really nice, expensive plant for super cheap if that person doesn't have a plant that's showing its full potential at any one time. And remember that this is not a science. This is not a synthetic or peer-reviewed system here. This is just cultivators, a cultivator's observation that has worked for me over and over, and that has been corroborated by some really great growers. So something I hope to codify in the future, maybe with some help. Next slide, please. All right. Have I talked about all this? Probably. So captive breeding, hybridization is how you interact with all of it, with trait knowledge. How I assess plants, what we'll be doing is we'll be going through each anatomical piece from the lid down, and that will include flowers and Phyllodia. And then I'll talk about a few of the phenological categories, a few of the phenological actions that the plants go through, their development over seasons. I don't think I'm going to touch on development of seedlings, and that's also quite a bit of fun. If you want to, if I'm sure that some of you do, imagine the possibilities when you combine a white top plant with a perp. Like, could you get a perp with a gigantic white lid on it? Surprisingly, probably not. You'd have to do that several times. But the point is, use your, become flexible, and through imagining each of these traits combined with each other. It is so beautiful to see that happen in real time. That's why I grow seedlings. Next slide, please. Let's start with lids. So I have to see my notes here. These are about all of the individual traits per lid that I could sum up. If someone else has an idea, I'd love to hear it. But generally, you know, you're going to be going by roundness, vaultedness, which is the not the gabling, which is in Oreophila particularly, the lid is almost curved, like almost like a conquistador's helmet. It has space within it that it bends over, and that on that axillary rachis there, surrounded by the veins at the middle. It's the midrib. So that's what vaultedness is. Gabling is a trait I see almost exclusively in flava. And if you see a leuco with gabling like that ain't leuco. That's a hybrid. Leuco doesn't have that. Very rarely, I don't think that I've ever seen leuco with anything like that. That's a cross. As you can see on the leaf behind it, it didn't have that gabling. You could have mistaken that plant for Leucophila, but it isn't leuco. It's leuco by Adrian Slack. And that's a seedling, so it looks a little different. Venation on the lid is very, very important. Very important. And Oreophila especially, I've noticed. These is the term adaxial? I'm not certain. But there are two artery-like veins that enter the lid from the sides, and they push up and they bifurcate like branches. And that's uniform in every single Sarracenia, from seedling all the way up to an adult plant. It's always there. And in Oreophila, you can trace that. You can they're very clear, but they're very minimal. Whereas in flava, it won't have the same expression. Oreophila var ornata might have it a little bit more, but you'll know if you have an ornata plant. That, coupled with roundness, coupled with vaultedness, the presence of a filament, those four traits there will differentiate Oreophila from flava. And that's a lot of traits. So it becomes very clear to me, at least, after seeing a lot of Oreos and a lot of flavas, what the difference is, because there are at least four traits in the lid alone that can tell me what one plant is and what one plant isn't. Whether or not they're species or subspecies of each other doesn't matter. The point is they're two completely different taxa. So there are actually more traits that separate flava from Oreo than you might expect.
Hey, Carson.
Yes.
There's a question asking what you mean by filament on a Sarracenia.
So I didn't really do that well in these pictures here, but if you look at that Oreophila picture, which is on the left-hand side of the screen, and you look at the very tip of the lid, that is where you would expect a filament to be in Sarracenia flava. Right there, a little bit farther up at the very tip, right there. Yep. So a filament can appear on flava that, and I believe hybrids with it and Leucophila are actually not too uncommon either. It's an inheritable trait. It's a heritable trait, all the way up to like an inch long. It's a very distinctive aspect of the plant. Not always there on flava, but usually it is. An Oreophila, it will either be not present or it will be such a tiny, tiny little recurved nub that it'll almost be a non-aspect of the plant. But that can be a reinforcing telltale difference between one species and another. Not always, but frequently. You know, if the thing has a long filament on it, it's not Oreo. And I don't think that it really carries over to Oreo very well. All right. If I answered everybody's questions, I think I did. Oh yeah, look, Jeff. Look at Jeff Green's screen. Look at that.
Put Jeff's screen up for a second.
Look at that big old filament. Look at how long that is. That's a flava filament. Thanks for illuminating that for me, Jeff. I needed that. See that? Yeah. Rubricorp or Dureens maybe by Don's Red Tube. Chocolate Top by Don's Red Tube. Look at the big old thing. So that's a very, very telltale aspect. In short, look at your lids. They're probably the biggest thing on your plant. Next slide, please. And that's an Oreo that Jeff Green's holding there too. Completely different. You can see the vaultedness and the curvedness of that lid. And the venation on the top is pretty significant. Okay, but that venation is very rare. And if you have that venation on something you suspect is Oreo, definitely send that to, if you're not certain, have a conversation about it with someone who might be. That's what I always do. So the operculum. So I discern, for the sake of I discern for the sake of this presentation, the operculum from the lid. And botanically, I don't believe that is a legitimate thing to do, but I do want to, I needed to break that up. The posture of the operculum is usually, or the neck is usually dependent on how heavy that lid is. But oftentimes it has its own personality going on with it. With flava, it can be either, if you're looking at the right-hand picture there, it can be either bent forward or straight upright. It's very desirable if it's upright. It opens up the plant. You can see on this hybrid in the middle that operculum is at a bit of an angle, and then the rachis, that mid-rib on the lid, which is basically the there's a barrier that defines the color delineation on the a color delineation that separates the operculum from the lid. I'm not quite sure where that lies, but that notch there is about not quite where it is, but it's associated with operculum structure intimately. The interior color is another trait that you can pass down from plant to plant. Not much. I don't usually use opercula for differentiation, but it's a trait I like to mention. Next slide, please. Okay, mouth shape and orientation versus nectar roll. You could have V-shaped faces. You could have round faces. You get something that looks like a toilet bowl. Something that looks like it's more upright. It's kind of weird looking. That minor thing there is actually at about a 45-degree angle. That's also a heritable trait, and you can use that to tell whether or not your leucophila is actually an areolata or a reedii, or whether or not that's, your plant is actually minor, or if it's rubra minor, or if it's flava minor. Using the shape or the orientation of that mouth will help solve that question. And then there's that perp thing on the side. Perp has a very usually a very conservative mouth shape. Usually, though, perp traits are all about the lip and the lid and the shortness. Next slide, please. I think we're talking about the nectar rolls. This is about the most interesting thing on the whole plant, is what its lips look like. Like that in the middle is crown of thorns, and that crenellation, which is an aspect that I included in the trait list that I used to discern where a plant came from, or what hybrids it was, what parentage it has, is about as desirable as things get, because the shape is just so unworldly. In the wild, believe it looks like it happens quite often. But in cultivation, only recently have we gotten beautiful shapes like that. And it's usually by combining one plant like Oreophylla with a leuco or a morii, which warps, not in a really complementary way either, completely warps the structure of the plant to accommodating not just shape but also texture. So crenellation might be a series of traits together. It might be one trait. It looks different from pitcher to pitcher on a single plant. One plant might not even crenellate one year, and the next year it might. Recognize that these traits are more complicated than I have listed them. There's of course a spout, just possibly a very distinct from crenellation because it's not a mix of traits. The color on the nectar roll, I mean, God, imagine it. And the width, of course, of the nectar roll has been very important in hybridization. Something that I found is that a lot of my plants that I was working with at Sarracenia Northwest had heritage from Sarracenia minor in them. And I found that out by first noticing that all of their faces had this narrow eyeliner-like mouth that was very pointed. Not just the mouth shape, but that nectar roll is so distinct and so uniform from pitcher to pitcher. Minor pitchers are very uniform. So that was a telltale sign that led me to realize, hey, these are hybrids with minor, not perp or rubra. And I was right, and it ended up being so. All right, we have a chat thing. What's going on? I think we're good. Next slide, please. I haven't been asked a question. So tubes. This is where I look at all those traits that I ended up finding here. Too many to go over. Color in the tube is about as important as it gets next to your mouth, right? on your plant. But there's so much going on in a pitcher plant's in the main body of the leaf that it can be very overwhelming. So I urge you to take your time whenever looking at your plant, not just judging right away that it's one hybrid or not if you don't have a label. But on this page, we have two species and a hybrid. The hybrid is the giant green sucker on the left, and that is a Micheliana. That's an anthocyanin-free Micheliana, and the other species are Jonesii and minor. Excuse me. In Jonesii, there's something fabulous going on with the upper part of the trap that it shares with Sarracenia Oreophylla. It's probably one of my favorite traits ever. Not only does it have a inflated throat on mature traps, not these, but it's translucent. And translucence is an aspect that I don't think very many people talk about when they consider non-leuco plants. This isn't areolation. This isn't light windows or light splotches that you see on the back of that minor pitcher there. In Oreophylla, Jonesii, maybe some Gulfensis plants, and maybe some Alata. Usually the Alata tends to be hybridized with something. But on Oreo and Jonesii especially, the top will have this translucent, almost daiquiri ice kind of complexion if it's green. That light will light up like a lantern and make it glow. And that trait not only can be used to differentiate, is this a and not usually with flava, because flava tends to cover that up. But I believe it applies itself to the very thin tissue. The thin tissue of Jonesii and Oreo is almost paper thin. It's almost like tissue paper. In fact, it's the texture of the tube that is probably the most telltale difference between Oreo and flava. Flava is very rigid, almost cardboard-like. It snaps when you bend it. Oreophila is very different. Oreo, and I have 30 Oreo clones, 30 of them. Every single one has a paper-thin trap that is prone to warping. And almost all of them have a level of translucence at the top of the trap that you'll never find in Sarracenia flava. And that's something I've never noticed in botanical treatments. Something that I'd like people to consider. Especially in hybridization. Less so in differentiating in discerning perhaps whether or not something has Oreo in it. Again, it's something that tends to get washed out. But hopefully you'll get one hybrid with an occurrence, but also just telling apart flava and Oreo. Another thing that Oreo has in the tube, as far as texture goes, it's rough, almost sandpaper-like. It's not quite pubescence, but it's getting there. Flava will never have that. Warei, albamensis will have that. Golfensis doesn't have that. Rubra doesn't have that. The albamensis complex, which includes Warei, was differentiated from the other rubras, not just based off of its flowers, but also by the texture of the leaves. And the texture of those leaves imply that it can't last through winter, which is true. They senesce. Jonesii a little bit differently, but all right. I think I've talked enough on this. Throat, inflation, rigidity, all of these aspects. You have to touch your plant. You have to interact with your plant directly to know your plant. You can't just look at it. You have to be rough with them too. You have to, pry them apart, break them, split them open, look at their innards, see what they've been eating. Prey are very different from minor to Oreophila. Very, very different. Minor eat ants, Oreos eat bees. So yeah, get in there and bust your plants up. Venation, like, my God, Allah dimensions. if you're looking at Allah or Allah, if your plant is supposed to be Oreophila, but it has one of these things, your plant's not Oreo. It's not. And I am blown away by how many times I've been given an Oreophila that has this enormous Allah on it. And if you look at the minor thing on the left-hand side here, that whole non-tubular strip of tissue next to the tube, that connects to the tube, that sail almost, that's Allah. That's not God, that's the Allah. and that there should never appear on some species. It should never appear on especially Oreo. All right, next page, please. I'm skipping over stuff here because there's way too much for me to cover in this very limited amount of time. God, we're already 40 minutes in. Phyllodia. That thing on the left-hand side is a Phyllodia from Sarracenia alata, a Phyllode from Sarracenia alata. It's very rare, but they do occur. If you have an alata that has Phyllodia on it, that does not mean that it's a hybrid. It can still be a species. Don't worry if you have that. Oreo Phyllodia, very distinct from Flava Phyllodia, which is on the side, on the right-hand side. Another thing that I've heard is that location forms of Flava have different shaped Phyllodia. Location form Flava from the Atlantic coast will have a curvature at the very top of their Phyllodia, a little hook. And Phyllodia from plants in Florida in Alabama will actually have a more straighter edge in general. If you want to tell the difference between individual flavoclones, location-formed plants, that is proven essential to people who are obsessed with them. And there are people who are obsessed with them, trust me. There's one guy who has a greenhouse for every single state locality collection on his farm. So this isn't, yeah, Mr. Gaelic. This is not an arbitrary distinction. Next slide, please. All right, flowers. I'd like to get to senescence here, so I'm just gonna mention that. The difference between if your Leucophila flower senesces from the bottom of the capsule up to the top of the capsule where it connects to the stem, odds are it's not leuco. And if it senesces from the top down, then it ought to be leuco. That at least follows the description of this species as understood in the wild. Does that mean that I'm going to go through my collection and mark everything else that might be a hybrid? Possibly not, but what it would do is it would allow me to infer that thing might have flava genes in it. So that's what I'm going to look out for, is flava things coming out of that plant. If I do crosses with it, or that thing came out of a flava dominant or a population with flava introgression or retrogression. And that can be a discerning feature possibly between populations or between population forms. On flava and on some species, am I getting that right? I'm not going to say that. On flava, at least, the flower will turn completely upright like a sunflower, some forms of it, and dehisce at that point, releasing the seeds. Petal length, scent, color, I mean, you name it. I don't think flava smells like cat piss. I think it smells like fruit. I think that, what's his face, Don Schnell was wrong. I think that it smells like fruit, not cat pee, but whatever. Next page, please. My nose is not broken, David. It's bigger than yours. It can smell better than yours. It's greasier than yours. All right. Growth point. In one species at least, the growth point has a tendency to submerge itself deep, deep into the soil, and that's minor. Sarracenia minor will do that. It might be for fire protection. It might be some other trait or some other reason that I'm unsure of. But when you see that, you can bet money that plant has had minor in its heritage. That on the left-hand side clearly has, but that's also a complex hybrid, so that tells me that trait can maintain through multiple hybridizations. Usually when that happens, the plant is not a heavy divider. Color. If you're an anthocyanin, if you've got an anthocyanin-free plant, your growth point will be green and yellow. It won't have a hint of red in it, where you'd expect it otherwise. And that's how you can tell plants apart when they're dormant, antho-free plants and otherwise. And then, as just a general example, it's a stuffed pot on the right-hand side for with red shoots coming from it. That's what you'd expect on any normal colored or most normal colored plants. Growth point color is very helpful if you have a lot of things that are that are dormant, and if you're able to find a pattern there, then you'll be able to. Sorry, the landline just rang. Then you'll be able to discern anthophyres from otherwise, or a set of non-anthophyres from each other by growth point color alone. Trust me, I've had to do this. Next slide, please. There we go. All right, so one other thing we're going to touch on here that I'd like to touch on. We're going by so fast. God, I'm sorry. I'm really forcing myself to ramble and skip subjects and skip the depth of all this. But I hope this at least introduces you to some ideas. Another thing here is whether or not your growth points overlap, and this will happen on some plants, no matter how big a pot you stick them in. Like rubras will naturally grow one growth point on top of another, whereas most flavas will grow in sort of a hand-shaped formation on the ramet. The ramet is the name for the whole plant, all the growth points included, I believe. Leo Song might be able to correct me on that, and I think that he's in the audience. But that's the only term I find myself using. You can tell plants apart this way. You can tell whether or not a flava is from north or south of the northern or southern part of its range this way, because this trait is inherited. This trait came about because of completely different growing conditions, evolutionarily. I wish I could augment this talk with evolutionary reasons or justifications, but I can't because I'm no botanist. It's also a heritable trait. And Phil Felici specifically selects for plants that divide uniformly and broadly instead of one on top of another. All right, and leaf peelability, that's another trait. If you peel the leaves off of 10,000 Sarracenias, you'll find that it is in fact a trait, and it is a discernible feature, a discrete feature per clone. Divisibility, whether or not they snap off, the presence of presence of scales. Scales are the hibernacula that enclose the growth point when the plant is dormant. Whether or not the size of those scales as they persist, not just the hibernacula, but they exist in between the leaves at the base on the rhizome. Not just the hibernacula, but they exist in the growing season as well. The length of those scales or the presence of scales at all is a heritable trait. The orientation of the rhizome, whether it's vertical or horizontal. Perps and sits grow vertically. Everything else grows horizontally. What if you have a rubra that grows horizontally? That ain't rubra. If you got a and then it's a question of, okay, can you tell the difference between different crosses of purpurea and Sarracenia? Clearly that's more complicated. But that feature is unique to Sarracenia and rubra, and upright, candelabraic almost rhizome. All right, I think I've talked about enough here. Next page, please. See where we're at. Root length. Thing with minor is that they have long freaking roots, and I found that out after I'm potting the sucker at Jeff's place. Those roots were like almost, I don't know, they were really long, man. They were really long, longer than proportionally than any other plant in the selection. And you look at that coloration, you look at that Allah. Is that perp in there? Yeah, there's perp in there. What else? Is it rubra? No, it's minor. And there's no areolation on there to tell you, but the texture and the coloration, that color scheme is minor. Same with the shape of the growth points, distinctly minor. Otherwise, had I not unpotted this thing, I probably would have just passed it off as a purpurea by rubra or something like that. If you look closely, the roots will tell you something. Next page, please. All right, oh God, here we are. So here's just a list of phenological aspects. Every plant has its own set of phenological traits, right? Every species has distinct phenological traits. Big aspect of a big difference between Oreo and Flava again is, the leaves senesce in Oreophila very early, whereas in Flava they maintain. foliar senescence like that has helped solve quite a few problems in the case of, is it a hybrid or is it a species between Oreo and Flava? We already talked about the shape of the mature ramette. Flower production, every plant has its own season, every species has its own season. And I'm sure that you can apply these traits to other genre of plants as well. With if you have a genus with like another 100 species in it, they're all from different parts of the world. They'll all come out of dormancy differently. You can extrapolate. These traits are not just for Sarrs. Anthocyanin-free traits, the whole thing doesn't have a single red coding gene in it. It bruises yellow and the petals might be white, I think. I don't like AF plants, so I don't really care, and that's all I look for. All right, next slide, please. I would like in the future to see all of this standardized, a standardized system, a standardized database where plants can be, where their traits can be listed, possibly in the same order that I've described here. Thanks, Patrick. But made me lose my train of thought, man. Jesus. Such a system already exists in the deconfusifier. You have a link to that on your document. I encourage people to make a whole spreadsheet for their collection. That would be a really nice idea. Record the ownership history as much as possible. This can all be very useful to our endeavors in conservation, horticulture, and ornamental horticulture and cultivation, etc. Listen to David. David is one hell of a devoted guy to our carnivorous plant hobby. He's very competent at designing web systems. So I think that, not just web, but apps and such like that. So I think that the future of our standardization of assessing would be held in the hands of people who can develop those computer systems. All right, next slide. Is that it? All right, my throat's getting hoarse. Let's get to questions. I've talked a lot.